Hello friends, today we are here to discuss the life cycle of rhizopus and the formation of the zygospore. In the previous lecture we have discussed about the vegetative structure of rhizopus. So we will just do a quick recap over here. The structure of the rhizopus was like these are the stolons, these are the rhizoids which grows underground, and these are the aerial sporangiophores which shows the formation of sporangium at the tip. Now you can see in this diagram that how the differentiation and maturation of spores occur inside the sporangium. So this is the peripheral portion, this is the central portion inside the sporangium, and in the peripheral portion it is a dense portion because the number of the nuclei as well as the cytoplasm is much more in this peripheral portion, and the central portion is less dense in comparison to this peripheral portion. And both the portion, the peripheral and the central part, it is demarcated by the formation of dome-like structure which is known as the columella, and the columella is formed due to the layer of the accumulation of the layer of the vacuoles. So in this peripheral portion, actually what happens, the cleavage furrows are formed. Due to the formation of these cleavage furrow, small fragments of protoplasm are formed, which inside those protoplasmic segments few nuclei gets accumulated, and these protoplasmic bits along with the nuclei get converted into the spores which form a fragile wall around them, and these spores they come out by the rupturing of this wall as you can see in this diagram. And when these spores will come out, the outer wall of the sporangium get ruptures and only the columella is left, and these spores are in the air and they just germinate on some suitable substratum and form the mycelium. Now in this case of Rhizopus stolonifer, this species is a heterothallic species. Now there are two terms we will see over here. One is known as homothallic species and the other is known as heterothallic species. Now we have to understand those terms. In the case of homothallic species, one example is Rhizopus sexualis, and the heterothallic species, the example is Rhizopus stolonifer. We are discussing the life cycle of Rhizopus stolonifer. That means we are discussing a heterothallic species over here. Now what is homothallic species and what is heterothallic species? In case of homothallic species, during the sexual reproduction or during the formation of zygospore, one mating type strain or one type of strains of hyphae are required, and in the case of heterothallic species, as we can see the Rhizopus stolonifer, the two types of hyphae are required or two opposite strains are required for the formation of the zygospore. Although these mycelium which are heterothallic or they show heterothalism, they are morphologically similar but they are physiologically different. So how these two different somatic hyphae are formed, we will discuss over here. But you can see that here is one diagram and here is one diagram. Both the diagrams are indicating the asexual reproduction and asexual reproduction. Only the differences you can see here I have written A minus, B minus, C minus likewise, and here A plus. This minus and plus strains, they indicate the strains. That is, this is the A minus strain and this is the A plus strain, or both strains are opposite. So in this case again the asexual reproduction is occurring. This is the mycelium, this is the sporangiophore, sporangia, and the spores is germinating over here. Same thing is here, but in this case you are observing that the spores which are formed of minus strain, and here the spores are formed of positive strain. So I hope that you got these 2 points very clear, that what is homothallic and what is heterothallic. So this is a heterothallic. Now what happened during the sexual reproduction, which occurs by the conjugation of two gametangium. So during the formation of zygospore or sexual reproduction, the two hyphae, the two somatic hyphae of opposite strain, one positive and one negative strain, come closer to each other. Now these two hyphae, they form a lateral branch, small lateral branch like structure, as you can see here and here. One is of red color. This red color is indicating positive strain and this blue color is indicating negative strain. Now these both structures which are known as lateral branches, they come close to each other and they enlarges in their size. Now this structure, it is known as the progametangium. Progametangium means the phase which is forming below the before the gametangium is progametangium. So this is progametangium, and during the formation of the progametangium, the tip swells and the lot of nuclei along with the cytoplasm get migrated toward the tip of this progametangium. After a certain time, this progametangium, the tip of the progametangium swells, and lot of nuclei along with the cytoplasm accumulated at the tip, and the rest of the portion of this gets separated by a septa-like or wall-like portion. Now this entire portion which is left here, left behind, is known as suspensor, and this is known as gametangium. So three terms: this is progametangium, this is gametangium, and the rest portion which is formed beside is known as the suspensor. Now what happens in this gametangium? Both the gametangium, they fuses with each other by the dissolution of this wall. So the wall of contact dissolves and the content fuses with each other, and the entire portion get mixed with each other, and the two nuclei of opposite strain get mixed up with each other, and these thick wall structure which is dark in color, black in color, having the outer wall body is formed, which is known as zygospore. So this zygospore is multinucleate structure. So after the formation of zygospore, the zygospore germinate. The germination of zygospore takes place, and the zygospore germinate by the formation of a small germ tube, which is also known as promycelium. So this germ tube or promycelium, which is the initial structure, but afterward it increases in the size and forms the sporangiophore. This sporangiophore forms at the tip a structure which is also globose or globular in shape, is known as the germ sporangium or zygosporangium, and inside this germ sporangium or zygosporangium, the germ spores are formed, which are mixture of positive and negative strain nuclei or spores. Now how this occur, we can just discuss like this, because inside this zygospore, the nuclei which are coming from this area, you have seen that this is red and this is blue. So this is the positive strain, this is the negative strain. So all the nuclei which are coming from this progametangium are of positive strain, and from this progametangium are of negative strain. So inside the zygospore, many groups of positive and negative nuclei are formed, which fuses with each other, and during the time of germination, meiosis occur inside the zygospore. And after the meiosis, 4 haploid spores are formed, and out of these 4, 2 are of positive strain and 2 are of negative strain. Thus in this, which is known as germ sporangium or zygosporangium, mixture of spores are formed. That is, few are positive strain and few are negative strain. Again, these will germinate and give rise to the positive strain nuclei or the negative strain nuclei, and which further forms the positive strain hyphae or the negative strain hyphae. In this way, we can understand that how positive strain hyphae and how negative strain hyphae are formed from this germ sporangium or zygosporangium, because it shows the presence of both type of spores, positive and negative. I hope you got these points very clear. Just remember the two terms, homothallic and heterothallic. Thank you friends. Now you can take the screenshot of this portion if you like.